Welcome back. I hope you are ready for some more differential action because we are going to be turning our attention away from the gram stain and the acid-fast stain and looking at two different differential stains, the bacterial endospore stain and the capsule stain. Now we're also going to show you a sample flagella stain. So be sure to find your way to the back of the room by the windows to see that example microscope with the flagella stain today. However, you will be performing the endospore stain and the capsule stain. So let's get jazzed for those and talk a bit about the background, beginning with an endospore. I like to say that when the going gets tough, the tough form endospores, and nothing could be more truthful. Endospores are the biggest badass structures out there. They are resistant to ultraviolet light, to chemical disinfectants, to all manner of chemicals, in fact. They are resistant to heat fluctuations, to desiccation. They allow a cell to survive when the going gets tough. They allow it to become metabolically inert, resistant, dormant, and to sit and hang out for a very long time. We'll talk about just how long in a moment, but I want to make note that there are certain genera of bacteria that are very good at forming endospores. These include two epic bacteria, Bacillus and Clostridium. Now, you really need look no further than a sample of soil to find these organisms. This is just a sample of dirt that we collected from the riverbed of Riverton, and so this would be a place where we could find endospore formers. Another place that we could find the epic Clostridium would be to look in the bottom of this Winogradsky column because Clostridium is anaerobic. So this is a soil and water sample from a riverbank that was made in the spring of 2015, so about a year ago, by a group of my students. This is called a Winogradsky column. You all are going to be building one during Lab 7, so you might want to start to think about where you're going to collect your samples. Down in the bottom, we would find the growth of Clostridium, an obligate anaerobe. So Bacillus and Clostridium are both incredibly infamous. Bacillus anthracis is responsible for the disease anthrax. We talked briefly about it in lecture. Remember the post 9/11 bioterrorist scares in which highly purified endospores were being sent around in the mail, and these endospores formed a white powder. When people received a package with the endospores and they opened it, if they inhaled those endospores, that caused activation and germination of the bacterial endospore. This caused something called pulmonary anthrax. Pulmonary anthrax is one of three types of anthrax, and it is the most deadly. It's also known as wool sorter's disease. There are two other types. There's a cutaneous anthrax. You can get in a cut in your skin. There is also gastrointestinal anthrax, but certainly the pulmonary anthrax was the one that became scary post 9/11. So Bacillus is an epic character. Clostridium also is. Because note, Clostridium botulinum and Clostridium tetani cause botulism and tetanus. And of course, those of you who are more in the trendy side of things will think about the fact that Clostridium botulinum is also the source of Botox. The botulism toxin is extremely powerful. In fact, if you just lick a spoon from an improperly home-canned good that hasn't been heated enough and it's contaminated with botulism, you will get sick. So it takes very little of this toxin to do a lot, and it takes very little of the toxin injected in the right place in your face to paralyze certain areas and keep you from having wrinkles. That's what Botox is all about. Now, this toxin actually inhibits acetylcholine at neural synapses, so it actually stops the signaling. On the flip side, Clostridium tetani and the tetanus toxin does the opposite. Super powerful signaling that can sometimes lead to contractions that are so violent that people would break their own backs. That's an epic image, an iconic image of those suffering from tetanus. So these two genera of bacteria are incredibly infamous. The third is not. It's kind of the odd man out. Spore Sarcina, however, is the only coccus that forms an endospore. So we know that these endospores are incredibly resistant and dormant. They develop in nature when moisture and nutrients are scarce, when the going gets tough. They're so resistant to environmental stresses that they can literally live for more than 100,000 years. And in fact, in 1995, there was an article published in Science magazine that is going to just knock your socks off. How many of you, okay, be honest, are fans of Jurassic Park? Yeah. What an epic old movie, right? Jurassic Park, you think, that's only in the movies. But guess what? In this article, they report on the extraction of an endospore from, you guessed it, 25 to 40 million year old Dominican amber, just like Jurassic Park. And they successfully activated, germinated, and saw outgrowth of this endospore. So perhaps endospores can stay tough for 25 to 40 million years. Let's look at the spore position within the sporangium. Now, that's a big mouthful. Sporangium is just a sexy term for the mother cell, the cell that is making the endospore and is going to die in the process of endospore formation. So it's basically setting aside its genetic material. When it does that, and we observe the spore, we might see that the spore sits in various different locations within the cell. This depends partly on the amount of time that's lapsed since the spore began to form, or also on the type of organism, the species that is forming that endospore. Some very commonly form this swollen sporangium, meaning that the mother cell that's making the spore actually becomes enlarged where the spore is because of the spore size and location. Sometimes we see endospores at the end of the cell, sort of near the end, and sometimes right in the middle. Now, this picture is a little bit confusing because it shows the spore as being green. And that's going to be true when we stain the spores specifically to make them green. And we're going to do that today. However, spores are not naturally colored. And in fact, as you might guess, they resist coloration. They don't want any dyes sticking to them. Part of their role is to resist chemicals in their environment. So oftentimes we actually see spores as looking like white sections inside of a colored cell if we only put in an average everyday cationic dye. Let's zoom in and look at the spore itself and talk about each one of the components of the endospore. This is zoomed in on just the endospore structure. Let's start on the outside and work our way in. On the very outside is this sort of thin, delicate covering called the exosporium. One immediately says to oneself, what? How can a thin, delicate covering be of any use to this endospore? It's really not. It's likely a relic of the formation, and it very rapidly gets lost after an endospore hangs out in that old Dominican amber. Not important particularly to function. Now let's zoom in just a little bit farther. Let's look next at this layer called the spore coat. The spore coat, this is where the amazingness happens. This is several thick layers of keratin. And you all, maybe you won't know this, but you're very familiar, in fact, with keratin. Keratin is the protein that's in our fingernails and in our hair that is very tough, very resistant. So it's impermeable to chemicals. If we go one layer deeper, we see another layer of protection. Remember peptidoglycan and what an amazing, rigorous molecule it is? It enables a lot of pressure to occur mechanically, even though it's very permeable, right? It's very permeable, but it also provides a lot of rigidity. So this is a thick layer of peptidoglycan. It may occupy as much as half of the spore volume. It's a lot of the spore. And it's providing that, very rigid layer. Next, we see the core wall, which also provides some protection for the core itself. And inside of the core, that's where the jewels are, so to speak, right? This is where the nucleic acids reside. It's where the ribosomes are. Everything that is actually needed to allow the spore to become viable again. And that's what we want to talk about now. But first, let's make sure we think about what it is about these structures that may protect the jewels of the core for just a moment. So really, this mechanism is still being studied. But what we do understand is that deep in the core where the DNA resides, there are protective DNA binding proteins. These DNA binding proteins literally wrap around the DNA in a very protective way. They're like a coat, like the lab coat that we wear in the lab to help protect us from outside forces. So these coating proteins around the DNA do the same thing, protecting from heat, radiation, desiccation. The cortex, remember that of course it provides rigidity around the core, but it does something else. If you think about when you put a popcorn kernel into the microwave and you heat it up, it pops, right? And that's because there's moisture inside of the kernel. The spore uses the cortex to remove or wick that moisture away from the core, keeping the endospore from popping when the heat comes, when it starts to heat up in the environment. Lastly, there's a couple of compounds within the spore that for a long time we didn't quite understand. Dipicolinic acid, complex to calcium ions, is always rich within an endospore. We know that that indicates that it must have some sort of function. And very recently, 2006, the Journal of Bacteriology published a great article showing that it seems that this dipicolinic acid complex to calcium ions seems to provide some heat resistance, though there's been some counter studies saying that there's still heat resistance that seems to be observed even in the absence. But it does seem to protect the DNA. It protects against oxidative damage, so remember hydrogen peroxide, very oxidatively damaging, and against desiccation. So here are some thoughts on why a spore is such a superhero. So now let's look at the process by which an endospore is formed. So in our Riverton riverbank sample, what happened with this soil is that we took it from an extremely lovely, moist, rich environment. We put it into this bottle and it's since been sitting here drying out. Of course, that's going to trigger those tough endospore-forming bacteria in this soil to begin to form spores. So what are the steps by which that occurs? Well, first off, the cell has to cordon away some of its nucleic acids. So it needs to make a copy of its genome. That is the first step. So you could say the DNA is duplicated. Likewise, you could simply say that a copy of the genome is made so that it can be set aside. In order to create the endospore around that copy of the genome, the inner membrane is going to need to infold, and so we're going to see that happening. So the cytoplasmic membrane folds inwards, and that is going to form something that we call the 4-spore septum. You can see that has occurred in these two events shown in this picture, and the endospore has begun to be formed. But of course, it doesn't have all of the layers that it will eventually get, and the next steps will enable that to occur. The membrane that exists around the original copy of the genome now reaches out and re-engulfs the endospore again. This is going to lay down yet another layer, and between those two layers, peptidoglycan will be synthesized, and that will create this very thick, very protective cortex. The spore coat can then be made and enriched with its keratin, and then finally the exosporium is formed, and the spore literally, and this is totes sci-fi, you guys are going to love this, the spore literally kills its mothership, right? It secretes lytic enzymes out into the environment to degrade and kill the original sporangium or mother cell. That leaves the endospore alone to hang out for maybe 20 to 40 million years old, or maybe even a little less if it's lucky and the environment becomes right before that. So what happens when the environment becomes right to activate and germinate that endospore? Well, let's think about the post-9/11 bioterrorist attacks. When an individual received a package with the white powder in it, and they breathed in those endospores, that's when activation occurred. Activation is typically triggered by a temperature change, and we know that going from that envelope environment into the lungs of the individual is going to cause activation. There's a big change in temperature. There's also a pretty dang big change in the chemicals, the local metabolites in its environment. So upon germination, that is when the cell begins to use those metabolites, food in its environment, it's got a lot of nutrients, a lot of rich nutrients within the lung. That's actually going to trigger it to swell. The spore coat is going to crack like that popcorn kernel, and the cortex is cracking, right? And metabolism and outgrowth are going to begin. The endospore is going to develop into a viable cell. And that viable cell, of course, can undergo binary fission. It can duplicate and duplicate and duplicate in that rich, lovable environment. And of course, that can allow it to cause pulmonary anthrax. So we recognize this as the process of activation, germination, and outgrowth. And we now understand the full cycle of endospores. We also understand that they don't just form willy-nilly. They form only when the going gets tough. So when the going gets tough, we need to find them to stain them. And that's why in our lab, we're going to be putting them onto a special medium. We got the recipe from the military. It's called a nutrient sporulating medium, or NSM for short. We're going to make the cells stressed out. We're putting them in an environment that is going to cause them to sporulate so that we can find endospores and see them on our stain. But of course, remember that the endospores are so dang resistant to staining that we're going to have a pretty extreme procedure in order to see them. So let's look at that procedure, and we're going to look at it in two ways. First, let's look at it in a large flow chart where we begin with the primary stain called malachite green. We're going to do that in the steam hood, and in a minute I'm going to walk over there to show you that steam bath. You're going to drive the malachite green into the spores. This is a water-soluble stain, so it doesn't have that much affinity for the cellular material, the lipid bilayers of the sporangium, but it does get driven into the endospore itself. We then simply use water as a differential step to wash away the malachite green from everything but the endospores. And then we can just use a basic stain, safranin, to color the vegetative cells. And they kind of end up looking a little bit pinky orange in the end. Let's look at this on a quick flow through from the virtual edge. So here's the endospore stain in action. You're going to walk over to the hood, and I'm actually going to show you where the hood is. So you're walking by the assignment chalkboard, and then you're coming on over here to the hood where you're going to put, as you see in this picture, you're going to put your cells onto your smear preparation onto the grate of the steam heat. You're going to add the malachite green onto a small piece of paper towel, and you're going to enable that malachite green to just be driven in by the steam heating. Once the steam heating is complete, and you can see it there steaming, getting that stain driven in, you're going to rinse it with the water, rinsing away the malachite green from the cellular material, from the mother cells. And from there, you're going to add the safranin counter stain, and you're going to color the mother cells, the sporangia. That is all there is to that procedure, and you should be able to see lots of really rad-looking endospores. They're going to look like little green guys, and let's pull out a picture and look at that. This is one of my favorite photographs that was taken by one of my TAs, and you can see the endospores are in green. They look like almost little green teardrops, and the sporangia, the mother cells, are in the kind of pinky orange color. So that's what we're expecting to find on your endospore stain. Now, the last differential stain that you're going to perform is the capsule stain. Remember that a capsule is a type of glycocalyx. So is a slime layer. We're going to be looking this time for capsules because we can actually see them in vitro on the bench top. So there's a really well-organized layer of material lying outside the cell wall, generally polysaccharide, carbohydrate, that is going to form a very thick protective coat around the bacteria. Now you can see a lot of reasons why a coat is protective. It can protect against external toxins, against bacteriophages, viruses that affect and attack bacteria. It's also great at protecting against our body's immune function. So bacteria that have capsules on them, when a white blood cell comes and tries to engulf this encapsulated bacterium, bloop, slippery little sucker, right? It just slips away from the grasp of the white blood cell. So as you might guess, this markedly increases the virulence of these bacteria. They're more likely to cause disease because our body can't cope with them very well. For example, Streptococcus pneumoniae causes most cases of bacterial pneumonia. We know that capsules have a great deal of water in them because they're polysaccharide, like dissolves like, lot of water retention. It's like spaghetti noodles, right? They take on a lot of water and they're slippery. And so we know that also will protect against any kind of hydrophobic toxin in the environment. So these are really good for a lot of things, but also if you think about the stickiness that's afforded by a capsule, it allows them to adhere to surfaces. And of course, if you search the internet, you can find many pictures of dental plaque if you just want to have that gross out factor. But Streptococcus mutans is at the base of plaque. Streptococcus mutans has a thick capsule. It adheres to your tooth enamel. That tooth enamel, of course, then is worn away by the lactic acid of the lactic acid bacteria, and that can lead to dental caries or cavities in the long run. That's why, of course, we try to remove our plaque when we go to the dentist or when we brush our teeth. So thinking then about these bacterial capsules, we can think about the fact that it may be kind of hard to stain them. They're all full of water, so we wouldn't exactly want to pass them over a Bunsen burner because there goes the capsule, no more capsule to stain. So we can't use heat fixation when looking for capsules. We have to take a different approach. So what we do is we try to stain the bacterial cell, and then we try to stain the background, and we leave the capsule unstained. So we use a little bit of a chemical approach to this stain, and we're actually going to use a really unique approach today. We're going to use milk, a milk culture. We're actually going to have a culture of Klebsiella pneumoniae resuspended in milk, growing in milk. And we're going to stain the milk, we're going to stain the Klebsiella pneumoniae cells, and we're going to leave the capsules unstained. Now you could do this in other ways as well. You could use a negative stain for the background. You could use a cationic stain for the bacteria, and that would leave, and then rinse the stain off of the capsule and leave the capsule unstained. The capsule ends up looking like a halo around the vegetative cell. And you can see that that's what we're seeing in this image. This is a sweet picture that was taken by one of my TAs, Rob Meyer, who interestingly is in dental school. Cool, right? So you can see that you've got a little halo around the vegetative cells in this image. So very cool white halo, darker purple cells. So very, very cool imagery of a capsule stain. Let's look at the process, the staining procedure, in a couple ways here. First, let's look at the formation of, the smear preparation. You're going to do the same thing as you would do from a liquid culture. You're going to take a loopful from your liquid culture of milk, put it onto your slide, and let it dry. You need to let it dry completely, and your TAs are going to help facilitate that process. Then you're going to hit it with crystal violet. Remember the cool cationic stain that we use for the gram stain? Everything is going to be stained purple. It's like stacks on stacks of purple, 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 right? Everything is purple. And then you're going to rinse with a copper 2 sulfate. That copper 2 sulfate is going to wash the crystal violet away from the capsule, leaving it coloring the background and coloring the cells. Now, I'd love to offer a little extra credit if someone wants to look up and explain why it is that copper 2 sulfate might rinse away the dye from the capsule, but not everything else. But we end up with this really cool image of dark cell, halo, dark background. Very cool. Let's take a look at this on Virtual Edge. You're going to begin your capsule stain by hitting it with crystal violet, coloring absolutely everything purple. You'll then just rinse that with water really gently, ensuring that your smear remains intact. You'll then hit it with copper 2 sulfate. What a beautiful blue color. That copper 2 sulfate sometimes, in fact, leaves the capsule with a light hue of that light blue color. And sometimes they just look more of a white color. And from here, you'll just rinse again with water. You don't want to blot this at all. You just want to air dry it so you've got your intact smear and you can actually see your very fragile capsules. That's all there is to it. So today you are going to finish the staining on your unknown. You're going to do an endospore stain. You've done a gram stain. You've done an acid-fast stain. You did a basic stain. You're going to finish off that unknown, and in lab you're going to write your unknown press release. So that should be a lot of wicked cool fun.